Hello, welcome to Zigmatech Learning Hub. I'll be your instructor for biology. For this class, we are going to be taking our exercises from the Examguide app. Now, if you don't already have this installed in your device, I would like you to download the app in order to follow along in this class. Now, Examguide is a leading educational app that helps students prepare adequately for various exams. Exams such as the UTME, the post-UTME, WEAC, GCE, IJMB, KCPE, JUPEB, Calbepedia. In the junior sections, we also have the BECE, we have the SSCE, and so much more. Now, you can download the app from www.examguide.com or you visit the Google Play Store to download. Now, please subscribe to our channel and turn on the notification bell to update yourselves on new videos that will be coming up. Now, if you're ready for this class, let's get started. Today, we are going to be looking at a topic called plant nutrition. Plant nutrition. Before we go into plant nutrition, there are several things you are expected to know as your specific objective. And let's take a look at some of these specific objectives. We have, by the end of this lesson, you should be able to define what we call photosynthesis and state the conditions that are necessary for photosynthesis to occur. Number two, you should be able to enumerate the stages of photosynthesis. Number three, you should be able to outline mineral required for plant growth and healthy development. Number four, you should be able to describe an experiment to test for starch in leaves of plants. And then number five, you should be able to describe an experiment to show that sunlight is necessary for photosynthesis. And also you should be able to describe an experiment to show that chlorophyll is also necessary for photosynthesis. And then finally, you should also be able to describe an experiment to test for starch, reducing sugars, protein, fats, and oils in food substances. Okay? Now, these are the things we're going to be looking at. Right. Now, but before we start up with the definition of photosynthesis, I'd like to take your mind back and refresh your thoughts or your memories on some things we have actually discussed concerning nutrition. Remember, we said that nutrition is the ability or the process by which living organisms obtain nutrients from food substances. And I also went ahead to explain to you that we have two types of nutrition. We said we have what we call autotrophic nutrition, and we said that autotrophic nutrition is a type of nutrition in which living organisms are able to manufacture their own food. They are able to manufacture their own food. And we said that all plants fall into these categories. And then we also talked about types of autotrophic nutrition. We also mentioned that we have two types of autotrophic nutrition. We talked about photosynthetic nutrition, which is also called holophytic nutrition. And then we talked about chemosynthetic nutrition. Now, under chemosynthetic nutrition, we said that living organisms manufacture their food, but they do not require sunlight to carry this out. It means that they synthesize or produce or manufacture organic materials, which are food substances, okay, from inorganic substances. They manufacture organic materials or substances from inorganic materials. Inorganic materials such as the ammonia, we have nitrites, nitrates, and so on. And examples of organisms that fall under chemosynthetic nutrition, like I said before, we have bacteria. An example of the bacteria is the nitrobacter. We also have the nitrosomonas bacteria. These are bacteria that mix or derive or manufacture their own food using nitrites or ammonia compound or ammonium compound. Okay? Now, the second type of autotrophic nutrition, like we said before, is photosynthetic nutrition. photosynthetic nutrition. And that is what we're going to be discussing today. We're all going to be looking at what is photosynthesis. Now, by definition, photosynthesis is the process by which green plants manufacture their food in the presence of sunlight, carbon dioxide, and water. Now, if you look at the text we have on the screen, they are highlighted in red. Now, those words that are highlighted in red are keywords. They are words when you are defining photosynthesis should be in place in your definition. I repeat, photosynthesis is a process by which green plants, manufacture their food in the presence of sunlight, carbon dioxide, and then water. You can see the chemical equation that represents photosynthesis. We have it as CO2, which is carbon dioxide, reacting with water to give rise to glucose, which is C6H12O6, plus oxygen gas that is given off as a byproduct. Now, it is also important for you to note that photosynthesis is an endothermic reaction. It is an endothermic reaction. In other words, heat is conserved, not given out, or released. Heat is conserved. Now, having gotten known this about photosynthesis, let's take a look at some of the conditions that are necessary for photosynthesis to actually take place. There are several conditions necessary for photosynthesis to take place. Now, if you had paid attention to the definition of photosynthesis, those words highlighted in red are some of the conditions necessary for photosynthesis to occur. An example remember we said green plants. Now, plants are green based on the presence of what we call chlorophyll. Now, plants cells possess what we call chloroplast. They possess chloroplast. And these chloroplasts actually give plants the green pigment that we see on the leaves. So leaves that are green simply means that these green plants possess chloroplast, which carries chlorophyll. Is that okay? So they possess chlorophyll. So chlorophyll is one of the most important conditions necessary for photosynthesis to occur. In other words, any plant that is not green cannot undergo photosynthesis. So photosynthesis is exclusively for green plants. Please take note of that. Only green plants can carry out photosynthesis. Number two condition necessary for photosynthesis to occur is there must be a presence of carbon dioxide. Remember that we said in the definition, in the presence of sunlight, carbon dioxide, and water. So carbon dioxide must be present. And number three, there must be the presence of water. There must be the presence of water. And then number four, there must be the presence of sunlight. There must be the presence of sunlight. Remember, as the word implies, photosynthesis. So it is, synthesis simply means to produce. Photo talks about light rather. So it means the production of food substances by light or with light, So sunlight is necessary for photosynthesis to occur. Please take note of these four important conditions. Now, these four conditions are not the only conditions necessary for photosynthesis to occur. We have other conditions like minerals. So minerals are required for growth. They are required also for development, which is one of the importance of photosynthesis. Number four, we have suitable temperature. The temperature must be suitable for plants to manufacture their food. And enzymes are also necessary and needed. When we start looking at the stages for photosynthesis or mechanisms of photosynthesis, you're going to understand that enzymes play a major role, or coenzymes as they call them, play a major role in the manufacturing of food substances. Now let's move to stages of photosynthesis. stages of photosynthesis. Now, before I go into stages of photosynthesis, you might want to know some of the importance of photosynthesis. I'd like to give you two importance of photosynthesis. Now, one importance of photosynthesis is that it releases oxygen to the atmosphere. If you go back to the definition we had for photosynthesis, you will see a chemical equation that was given beneath the definition of photosynthesis. And in that definition, we saw that one of the byproducts was oxygen gas. So it means that it releases oxygen to the atmosphere, and oxygen to the atmosphere helps in respiration in other living organisms. So that is one of the most important one of the most importance of photosynthesis. It releases oxygen into the atmosphere for respiration. Number two importance is that it helps to purify the atmosphere. Number two, it helps to purify the atmosphere. It is said that the more, it is said if you go through carbon's oxygen balance, based on carbon oxygen balance, the more carbon dioxide is added to the atmosphere, the more the atmosphere becomes polluted, if I'll put it that way. The more the atmosphere becomes impure. So an increase in carbon dioxide level has, will I call it an environmental hazard it creates or it brings about, okay? For instance, carbon dioxide is a greenhouse gas. And as a greenhouse gas, it can lead to global warming. So when you have carbon dioxide in the atmosphere increased, all right, it brings about global warming. And of course, there are several effects that will come as a result of global warming. All right, so adding more oxygen to the atmosphere helps to purify the atmosphere, thereby reducing the level of pollution in the atmosphere. And then number three importance of photosynthesis is that one of the products of photosynthesis is glucose. And this glucose is used by other organisms as well as the plants for food. So these are three important major importance of photosynthesis to the ecosystem. Please take note of that. Three importance of photosynthesis. Now let's come back to what we're about to look at, which is stages of photosynthesis. Stages of photosynthesis. Now, photosynthesis in plants, or mostly in green plants, is carried out in two different reaction stages. Two different stages. The first stage is what we call the light reaction stage, and the second stage is what we call the dark reaction stage. So we have the light reaction stage, and then we have the dark reaction stage. Now, let's take a look at what light reaction stage is all about. Now, there are several steps you should take note of in the light reaction stage, and I'm going to list out some of these steps. Now, number one, the first step is that in the light reaction stage, okay, first of all, note this, as the name implies, light reaction stage, it simply means it occurs during the day, or it occurs in the presence of sunlight. So light reaction stage begins with the use of sunlight. Now, the energy that comes from the sunlight, that's the first stage, the energy that comes from the sunlight is being trapped by this chlorophyll. Remember, we said that green plants possess chlorophyll, or they have chlorophyll. So this energy is being collected or trapped by chlorophyll. And that energy that is trapped by chlorophyll is used to split water. Water is splitted into hydroxyl ion and hydrogen ion. I repeat, the energy from the sun is trapped by chlorophyll, and that energy is used to split water into hydroxide ion or hydroxyl ion and hydrogen ion. Now, this process is called photolysis of water. Remember, photo talks about light. Lysis or lysis, L-Y-S-I-S, talks about splitting of water. So photolysis of water is simply the process by which water is splitted by light. Okay, so that's the first thing that happens, photolysis. Now, after the light is being splitted into hydrogen ion and hydroxyl ion, the hydroxide ion undergoes further reaction. That's the second stage. It undergoes further reaction to give off water and oxygen gas. So the oxygen gas that is usually given off during the process of photosynthesis comes from the further reaction of the hydroxide ion. Do you get that? Now, the third stage is that hydrogen ion then reacts, listen, with a coenzyme, and the coenzyme is NADP, which is nicotine amide dinucleotide phosphate. So it reacts with NADP to form, and it reduces it from NADP to NADPH plus. All right, NADPH plus. So the coenzyme is reduced by the reaction of the hydrogen ion with the coenzyme. Now, once this thing is done, energy is being released. Energy is produced. That energy that is produced is in the form of ATP. So these are the stages that takes place during the light reaction stage. I repeat again, in the light reaction stage, the first thing that happens is photolysis of water. It simply means that the light energy, or the energy of the sun, or the light of the sunlight, is used to split water into hydrogen ion and hydroxyl ion. The hydroxide ion undergoes further reaction to give off oxygen as the byproduct. Then the hydrogen ion reacts with a coenzyme called NADP and reduces it to NADPH plus, and then energy is produced in the process. So this is what happens during the light reaction stage. Now, the next stage is the dark reaction stage. Now, what happens during the dark reaction stage? Now, in the dark reaction stage, it occurs at night in the absence of sunlight. Occurs at night in the absence of sunlight. Now, what happens is that this NADPH and the ATP, which is the energy, moves into the dark reaction stage. The hydrogen ion reacts with now carbon dioxide that is present. So the carbon dioxide comes into the reaction during the dark reaction stage. Okay, so the hydrogen ion from the NADPH reacts with the carbon dioxide and with series of chemical reactions. Series of chemical reactions. Each of these stages, series of chemical reactions, at the long run, with the energy that is being produced, glucose is formed, or a 3-carbon compound is formed. All right, or a 6-carbon compound is formed, which is a simple sugar. So these are the processes or stages that occurs during photosynthesis. I repeat, in the dark reaction stage, the hydrogen ion from the that reduced the coenzyme NADP, and the energy that is produced during the light reaction stage moves into the dark reaction stage to react with carbon dioxide. And each of these stages are powered by the energy, which is ATP, and several reactions takes place to give rise to a simple sugar. And that simple sugar is the byproduct of photosynthesis, as well as oxygen gas that is given off during the light reaction stage. All right, now move ahead. We have evidence of photosynthesis. There are evidences of photosynthesis. One of the major evidence of photosynthesis, or one of the evidence that proves that photosynthesis has taken place in green plants, is the formation of sugar. So in other words, if we want to check if a green plant had undergone photosynthesis, it simply means we have to test for starch or sugar. Now, that sugar that is being produced, why I said starch, is that the sugar that is being produced, part of it is used by the plants for food. Now, the remaining part of the ones that are remaining part that is not used by the plant, that is the excesses that are not used by the plant, are converted into starch and other forms of food and transported by the phloem to other areas or parts of the plant where they are stored. To other parts of the plant where they are stored. And the process by which the phloem transports manufactured food from the leaves to other parts of the plant is what we call translocation. Translocation. So translocation occurs after the plant has made use of the food it has manufactured. The excess of it is being stored in several parts of the plant, and they are stored mainly as starch and other forms like fats and oils and proteins. Okay, so one of the ways or evidence or ways to prove that a particular plant or green plant had undergone photosynthesis is to test for starch. All right, now there are mineral requirements or minerals that are required by plants for healthy growth and development. All right. Now, these minerals include, they are divided actually into two main groups. We have what we call the macronutrients, and then we have what we'll call the micronutrients. Now, macronutrients, another name for macronutrients, they are called essential nutrients, or they are called major elements. Essential elements, major elements. Now, the reason why they are called major elements or essential elements is because they are required in large quantities. They are required by plants in large quantities. Now, some examples of these macro or essential elements include, we have calcium, we have potassium, we have phosphorus, we have nitrogen, we have hydrogen, we have carbon, we have sulfur, and so on. These macronutrients or essential elements are needed in large quantities for plants' healthy growth and development. And then we also have the micronutrients. Micronutrients. They are also called trace elements or non-essential elements, or you can call them minor elements. Now, the reason why they are called non-essential is because they are not required by plants in large quantities. They are required by plants in small quantities. Examples include boron, we have molybdenum, we have chlorine, we have cobalt, we have copper, we have manganese, and so on. They are required in little or small quantities. All right. Now, the next thing again, if you recall, we talked about evidence that a particular plant had undergone photosynthesis, and we said one of the ways to test or prove that a plant has undergone photosynthesis is for us to test for starch. Now, there are several procedures for testing for starch, and please take note, anytime you are carrying out any biology experiment, that means you are carrying out the experiment, you're doing it yourself, please take note, you are to actually document. Okay, put a record down of things you are doing. And how do we put these records down? How do we put these records down? You start with the aim. The aim talks about the objective, why you are doing the experiment. For instance, in this case, testing a leaf for starch, we can say one of the aim is to demonstrate that a leaf had undergone photosynthesis. Or you can also say the aim is to test a leaf for starch. To test a leaf for starch. That is the aim. Now, the next thing is the apparatus or materials. Now, the apparatus or materials are the instruments or the materials actually you are using to carry out this experiment. As we can see on the screen, there are several materials we have there. We have a Bunsen burner, we have a leaf, we have ethanol, we have beaker, we have test tube, we have a slide, okay, sorry, not even a slide, a white tile, and so many other things that are necessary to carry out the experiment. So please write out your materials. And then number 3 thing to do in your records is that you should write out the methods or the procedure you went through, the procedures that are involved while carrying out the experiment. And then we also, after the procedure, you have the observation. What did you observe? What did you see? What are the things that came up after you have gone through the procedures? After carrying out the experiment, what did you observe? And then finally is the conclusion. Your conclusion, please take note, your conclusion must be, well, I call it synonymous, or must have a relationship with the aim, or your conclusion must interpret or link with the aim. Please don't forget that. So let's take a look at testing a leaf for starch. Now, the thing you do is that you pluck a leaf out from a plant that has been exposed to sunlight for, let's say, up to 2 days or even up to 6 hours or so. Now, when you pluck out that particular leaf, okay, you get the leaf, you get a beaker of water and then heat it. Now, once the beaker of water is boiling, you insert the leaf into the boiling water or into the beaker containing boiling water. You insert the leaf and leave it there for close to 6 minutes or more, okay? 6 minutes or more, let's say 10 minutes, you allow it to boil. Now, one of the reasons is to kill the cells and burst the starch grains open. To kill the cells and burst the starch grains open. So that's the major purpose or reason for boiling the leaf. Now, after boiling the leaf for that 4 to 6 minutes, you bring out the leaf and also take the leaf and put it in a test tube containing ethanol, 70% ethanol. Put it in the test tube containing 70% ethanol, and the ethanol must be a bit warm. Now, you leave it there. Now, one of the purpose for putting it into the ethanol is simply to decolorize the leaf. To decolorize the leaf or to remove the chlorophyll. Okay? Now, after that is done, you put the leaf in a hot water, not boiling this time. Please take note. The first one must be boiling. You put it in a boiling water. Then in the second one, you just put it in a hot water or a warm water. Now, once you put it there, the reason for putting it in a warm or hot water is to soften it. Okay, to soften it. Then after you have done that, you bring it out from the hot water and then put it on a white tile. You put it on a white towel and add few drops of iodine. If starch is present, the leaf will stain blue-black. It will stain blue-black. Now, if starch is not present, the leaf will not stain blue-black. So the blue-black coloration is an evidence that the leaf had undergone photosynthesis because of the presence of starch in the leaf. Is that okay? Now, the next thing experiment to look at is experiment to show that sunlight is necessary for photosynthesis. Sunlight is necessary for photosynthesis. Now, what you do is that a plant, okay, that is exposed to sunlight for quite a number of time, what you do is that you don't pluck out the leaf first. Don't pluck it out. Leave it where it is attached to the plant. What you do is that you take a black paper strip and cover a part of the leaf. Cover a part of the leaf and clip it to hold it firm. Now, allow the leaf to stay all through the day till the following day. And then when it gets to the following day, pluck out that particular leaf and remove the paper, the black paper strip, remove it and test the leaf for starch. Now, once you do that procedure and test the leaves for starch, you will discover that the part that is not covered with the black paper will stain blue-black when you add iodine solution. But the part that is covered with black paper will not stain blue-black. It will not be affected by the iodine solution. It will not be affected by the iodine solution. The reason is simple. That black paper strip prevented sunlight from getting into the chlorophyll for photolysis of water to take place. So photosynthesis could not take place because that particular part of the leaf has been deprived sunlight, We're going to be looking at another experiment, and the experiment is to show that chlorophyll is necessary for photosynthesis to occur. Now, in this particular experiment, we will be making use of a variegated leaf. Now, a variegated leaf is actually a leaf that has patches. That means the leaf have green and white patches. It has patches. Now, in these patches, so in some plants, you have green and yellow. Some of them is green and white and so on. But there must be a green color in the plant or in the leaf of the plant, okay? Now, how do we carry out this experiment? Now, you get the variegated plant, let that particular plant be exposed to sunlight for quite some hours. Now, after that, pluck a leaf, a variegated leaf, and then test that variegated leaf for starch. Remember the process we said about testing for starch? You insert it in boiling water, you remove it and insert it in 70% ethanol. You remove it and insert it in a hot water, and then you put on the tiles, and then you put few drops of iodine. Now, once you put the few drops of iodine on a variegated leaf that you're testing for starch, the green part or patch of the variegated leaf will be affected by the iodine solution. Hence, you're going to see a blue-black coloration. But the part that is not green, that is the non-green patch of that variegated leaf, will not be affected by the iodine solution. You won't see a blue-black coloration. It might be colorless, okay? So that is also an evidence to show that chlorophyll is necessary for photosynthesis to occur. Now, the next thing we're going to quickly look at is test for starch in food. Now, how do we know that a particular food has starch in it? Though this particular discussion is supposed to be fitting under animal nutrition. But I just want you to understand this. How do we test for food or test a particular food for starch? Now, one of the simplest way to test for starch is to use the iodine solution. Now, also please remember, like I said before, that whenever you are carrying out any experiment, your records are very important. Your records are very important. You start with the aim, you go to the apparatus or materials, your methods or procedures, observation, and then your conclusion. Now, when you want to test a particular food for starch, what you do is this: get the food sample, okay, probably in a solution. Let the food sample be in a solution preferably. Now, what you do is that you put few drops of iodine solution, maybe two or three drops of iodine solution. When you add those few drops of iodine solution, that food sample will turn blue-black. Now, if it turns blue-black, it means that the food in question or the food sample has starch in it or is a starchy food, okay? Now, the next one quickly we're going to look at is to test for reducing sugars. Now, one way we test for reducing sugars is the Benedict's test, using Benedict's test. Now, what we do is this: we take 2 cm cube of the food sample, which is the reducing sugar, and put it in a test tube. Now, once you do that in a test tube, you add equal amount of Benedict's solution to it. You add equal amount of Benedict's solution to it, and then you heat it in a water bath. You heat it in a water bath. Now, if when you heat it in a water bath, the solution turns brick red, it means that that solution contains reducing sugar, or that solution is a reducing sugar. And then also to test for proteins. To test for proteins in food substances, what we do is that we take a particular food sample and put it in a test tube. You add few drops of dilute copper sulfate solution, and after which you add few drops of dilute hydroxide solution. Now, once you add the few drops of dilute hydroxide solution, you will see a purple color coming out or appearing in the food sample solution. you will see a purple color appearing in a food sample solution. That is, if the food sample solution has protein in it, or if it is actually a protein. All right. And then finally, to test for fats and oils in food, the first thing you do is you crush the food sample and put it in a dry test tube. After that, you add ethanol to about 2 cm³ above the level of the sample and shake thoroughly. Now, after that, allow the solid to settle about 3 minutes to allow the fat to be extracted. After extracting the fats by decantation, or by decanting the ethanol into another test tube, add 2 cm³ of water to the second test tube. All right. Now, once you add that 2 cm³ to the second test tube, if you notice a white cloudy emulsion, it means that it means that fats and oils is present in that particular food sample. It means that fats and oil is present in that food sample. Another way also to test for fats and oils in food is to use what we call the translucent test. Now, the translucent test is that you crush the food sample you want to test for fats and oils, and then make a smear on a filter paper. You make a smear of it on a filter paper. When you hold that filter paper against a light source, if that filter paper, that particular place you made the smear, is translucent, it means that that food sample you crushed is fats and oils. Now, this brings us to the end of this particular topic, plant nutrition. But before we go, let's take 1 or 2 questions from the Examguide app, SSCE. All right. Let's just very few questions. We're looking at random questions picking from different years. Now we have here, they said the major role played by accessory pigments during photosynthesis is to, that is, the major role played by chlorophyll is to what? A, is it prevent chlorophyll from absorbing unnecessary light? B, is it trap light energy that chlorophyll cannot absorb? C, is it add color to the plant but to trap light energy? C, D, is it transfer energy from chlorophyll to other pigment? And the correct answer should be B, trap light energy that chlorophyll cannot absorb. Then we also have this particular question. They said the process by which plants manufacture food from carbon dioxide and water using energy from the sun. Of course, the correct option is C, which is photosynthesis. And then finally, question 4. Look at this. Which of the following solution is suitable for removing chlorophyll in green plants? If you could recall, when we were looking at testing a leaf for starch, we said that ethanol is used to remove chlorophyll or to decolorize the leaf. Now, B is Benedict's solution, it's not ethanol. C is Million's reagent, it's not ethanol. D is Felling's solution, it's not ethanol. The correct option to this answer is A, methylated spirit. Thank you for participating in today's class. You can practice more questions using your Examguide app. The app scores and gives a detailed explanation of all the questions at the end of your practice test. You can also learn particular topics of interest with different modes like study mode, mock mode, and even practice mode. It also has other features that makes learning very fun. Now, it is a must for all serious students. Download from www.examguide.com if you don't have it yet. See you in the next class. Don't forget to subscribe to our channels, hit the notification bell, and share the videos to your loved ones and friends that will benefit from it. Bye for now.